Sorry for the inconvenience. Good afternoon to all. We start with the second session of day 3 offline AICTE sponsored ATAL 1 week online and 1 week offline faculty development program on research and innovation ecosystem in academics need of the hour. I request Principal Madam to felicitate and welcome Dr. Pradip Chaudhari with a sapling. I request Sir to occupy the chair on the dais. It is my pleasure to introduce today's resource person, Dr. Pradip Chaudhari. Dr. Pradip Chaudhari has completed his post-graduation degree in veterinary science from Bombay Veterinary College, Mumbai. University of Mumbai has awarded Sir a postgraduate diploma in medical radioisotopes and technology as well as a doctoral degree in applied biology. After completion of postgraduate studies, Dr. Pradip Chaudhari joined Bhabha Atomic Research Center in 1994, where he worked in the field of basic and applied nuclear medicine research. During his BARC assignment, he worked exclusively on animal model development for cancer, osteoporosis, and arthritis. He has also explored application of nuclear medicine diagnostic procedures for veterinary patients that have laid a foundation of dedicated veterinary nuclear medicine center at Bombay Veterinary College. He has played instrumental role in biological evaluation of various PET and PET diagnostic and therapeutic radio tracers. He has initiated a nuclear imaging for large as well as small animals. The work he carried out for his PhD involving biological evaluation of therapeutic agents for rheumatoid arthritis and bone pain palliation has translated for clinical application in humans. He moved to ACTREC, Tata Memorial Center in the year 2007 to establish a preclinical imaging facility and comparative oncology program. He is actively involved in preclinical PET, SPECT-CT imaging and exploring the utilization of spontaneous cancers of PET animals for basic and translational cancer research. Dr. Pradip Chaudhari has been awarded Boys Cast Fellowship, that is Better Opportunities for Young Scientists in Chosen Areas of Science and Technology Fellowship from Department of Science and Technology to pursue work in radiopharmaceutical research involving animals with spontaneous cancers. Sir is a founder member and past secretary of Laboratory Animal Scientists Association of India, which deals with laboratory animals used in biomedical research. Sir is also founder secretary of NNPR Veterinary Oncology Foundation. He has several publications and book chapters to his credit in various areas he has worked in. He was a part of several committees in past and presently serving as a member CPC SEA. Besides his academic career, he is an avid runner, nature lover and amateur photographer. Presently, Sir is a scientific officer at ACTREC Tata Memorial Center, Mumbai. With this brief introduction, I request Dr. Pradip Chaudhari to start with his presentation and share his expertise on preclinical, non-invasive imaging for cancer research with the audience.
Good afternoon, everybody. I'm really honored to listen to my own CV. Yeah. And there is always scope for refinement in everything. While I was just listening about myself, there is a lot of scope is there to refine the things. and present you very appropriately in front of the very unknown people. Yeah. And what we do in a research is the refinement. Research is always refinement of the things and adding new to the knowledge. Yeah. So in today's lecture, before I start, I must thank to Dr. Supriya for inviting me here, giving me this opportunity to interact with you all FDP ATAL faculties who are very keen on learning the things and very enthusiastic in taking, own their field in a very higher levels. Yes. So thank you, madam and your team. They were all coordinating with me, my time and everything. So it was really nice to visit this college. I visited earlier also, but always it's enriching me to visit this whole institute. I'll speak about the preclinical non-invasive imaging. Can I have lights on this side? Yes. Perfect. Yeah. So preclinical non-invasive imaging. We have just now seen that just the day before yesterday there was a notification by government that few drugs will be removed from the list of essential drugs. And in those, the list is like, we are very often take ranitidine. Yeah. And that has been removed now. Why? Because it has some carcinogenic kind of, potential. Yeah. So continuous or over excess use of these drugs might cause the cancer. So how important is, to do all those tests in animals? So those tests has to be very robust here. So what we do, we'll have some review about the background of this field as well as non-invasive imaging. Yeah. So use of animal research, I don't have to really tell much about this because you all are aware that normal physiology, normal gene function, human diseases, everything, if you want to learn, we need to have a biological system for that. Because in our in vitro system, what happens? We are always using a compartmental based kind of analysis, where only single cell is there or multiple cells are there. But when it goes for a complex system like human beings or that drug, it's very complex. Yeah, and we really don't understand much about it, how they interact with each other. Yeah. And for that, whatever our model system is there, it should be very fitting to the purpose what you are looking into. Yeah. So many drugs they are there which are very promising, and at the end we find it doesn't really work the way we have expected. So these are the basic tests usually we do it in animals. Yes. And you all are aware about the thalidomide tragedy, which has revolutionized the whole FDA regulation and all. And there is a one story also there was a one female who was leading as a FDA chief director from US. She refused that preclinical data of thalidomide. And she has not given that company to use that in United States. Yeah. So all pharma lobby was pressurizing her that she is more arrogant, she is very bureaucratic, and she is not giving us permission. It's such a promising drug which is already used in Europe, and she is not allowing us to do it in USA. And later we have found that what has happened to that drug, what it has caused. So she has saved so many human beings, from that her decision, and that was just because of her sheer preclinical data. She felt that this data is not enough, and that was the reason she took that decision. So there, after that, so many tests we do it for licensing of the human medicine as well as veterinary medicines also. And in this toxicity and teratogenicity, carcinogenicity, these are the studies that added much later now. Carcinogenicity is very highly 2-year study is there, and you look for different kind of benign or malignant growth in those animals or deaths in those animals. So if you go back to the history of animal experimentation, it's not just we started 50 years back or 100 years back. Before that, everybody was curious about knowing about the human body and how that time, it was difficult to, do the dissections and all those things. So they used to do it on carcasses. Yeah. Even human cadavers were abandoned by religious reasons for dissections. So animals were there basically. They used to think that okay, the animal blood is also red, human blood is also red, so there is similarity, and that's how they used to open up, dissect the animals. There was no anesthesia during those days. Yeah. So it used to be a very difficult, tough kind of a task how to do such studies. But when a group of like-minded people comes, they find some solution to it. Yes. So similarly, you can see here they are dissecting the body of a dog. Yeah. And they are trying to learn various organ system. So Galen was one of the Greek physician and who has done very systematic studies on the animal physiology. Yeah. He is also known as the father of vivisection. So he has done so many different procedures in animals and he has recorded so meticulously that those were helpful for several 100 years after that. You can see here the pig is being dissected and all people are curious about what is there in their body, how the heart looks like, why he died, all those sort of things are there. So then if you consider the statistics about these animals, in that race now you can see that different varieties of animals are used. But if you see the mouse and rat, their percentage is very high. And there are the reasons are there because since beginning we are using the very small kind of a system that mimics the whole complexities of the human system. And they are very easy to breed in the small environment. They are prolific breeders. If you see the gestation period of mouse is just 16 to 18 days. It's the smallest gestation period of mammal here. So such characteristics have made them to be used for research. And then the various strains have been developed. And there are now more than 110 strains are there of mouse. Yeah. Similarly, the rats also have a different varieties of strains. Other animals used are large animals like dogs are used, chimpanzees are non-human primates. They are also used, but now their use has been come down significantly because of several ethical reasons. And if you see this graph, this is from 2014 to 2018, the graph has come down. Almost 42% decrease is there in number of animals used in research. And one of the most important factors for that, as we are advancing our knowledge, we are using very advanced refined animal model system. Simultaneously, we are using the very advanced techniques for this. And that's why the number of animals used in research has come down. And adding to it is the ethical reasons for that. Now everybody is very conscious about how many number of animals I am going to use, what is my study design, whether statistically that will give me a proper data or not. All those questions I had discussed at the beginning of designing the experiment. And that has reduced the whole number of animals in research. In India, we really don't have much data about it, but as experience I can say that, or even madam can say that, when we were doing our research as a student, number of animals used by us and now how people are, our approach towards using them has significantly come down. At the same time, I would like to just, how many of you feel that the animal should be used in research? Almost all. Who says that animal should not be used for research? Anyone? No one will be there because we all are scientists. We are pro research. So we always feel. But there are many people that think that okay, no animal should be used for research. Similar people are also there, or mentality is also there. But just in this number, I would like to state more than 10% animals we slaughter for our food purpose. Yeah. So very few animals are now being used for research. If you compare the data earlier and now, there is very significant decrease is there. So the advanced techniques as well as refined models, these are the most important things. So in advanced technique, imaging modalities have came up in last 2 decades very significantly. And the reason for that, non-invasively you can understand or visualize what is happening in the body. So non-invasively it happens means that animal you don't have to really dissect. And so the number will be very less. But this modality is not a single modality, there are multiple modalities are there, and on the basis of their kind of information they give, their classification is there. Like some are there anatomic modalities. So in anatomic X-rays are there and CT is there. CT basically the X-rays done in a 3-dimensional way, 360 degrees. Then MRI is there, which has also anatomic as well as the physiologic component. Then in metabolic imaging there is a Serenca, optical, PET spec. Even as our science is going much more, medicine is going much more humanized or customized, and at molecular level, we need to have tools which are looking into or giving the information at the molecular level. So we have the molecular imaging modalities, ultrasound is there, functional MRI is there. So these are the various kind of modalities. So it is very difficult to say which modality is best. So that will be always a kind of a question, which modality is best for what reason or what question you are asking for. So on that basis this will be decided. Main difference between X-ray imaging and gamma ray imaging, like which is used in a PET radionuclide imaging, is that in gamma rays the animal whole becomes the radioactive one because we are administering the radioactivity in the animal. And in X-rays what happened? We all have undergone at least a single X-ray in our lifetime. Yeah. So we just stand between the X-ray source and the detector system. That's it. So we don't get any kind of an injection of radioactive material. So X-ray is very straightforward. So here the animal becomes radioactive or subject becomes radioactive. Yeah. So this is a basic difference. And also there is a difference between gamma rays and X-rays in their origin of starting point. So X-rays basically these are the rays starts from the outer orbits and gamma rays starts from the nucleus. So this is the basically a difference. So why small animal imaging? As we have seen, we want a model system for everything because we want to have mimic the system, complex system involved. And this is the main reason. In vitro is not equal to in vivo. So whatever you see in a petri dish, it is very difficult to reproduce in large animal system or multicellular system. Besides, we cannot really see the whole animal in one scan in many modalities here or in many assays. Then rapid in vivo screening, because now everybody working in a kind of a platforms, we are having a big data analysis, very different arithmetic or algorithms are there, and so many new targets we are identifying every time. And every target we feel that they have some potential. So there has to be a rapid throughput, high throughput screening also about those kind of molecules. So we need to have really a very rapid in vivo screening of those drug molecules. Then that whatever technology you are using, that should be non-destructive. Then only you can do a repetitive studies in that. And that is why the imaging comes into a picture. And that technology should provide a bridge between animal and human studies. So many assays are there, they are independent, they are not being done in human reality, in routine practice. But in this preclinical imaging, there are some tracers are there, they are also used in humans, same way. And the same tracers we are using in animals. So they form a very solid background or solid reason to take your outcome of that study further to the human trials. Yeah. New drug discovery have gone to a molecular level, so you have to have molecular kind of a insight of those studies. And translation of drug discovery from mouse to human. And that is why we need to have a dedicated kind of animal imaging instrumentation. because we are trying to compare or mimic a system of 25-gram rabbit with a human system of kind of 75-80 kg. Yeah. And we want to scan a mouse like a man at that resolution. So we have to have a dedicated system. We can also use the large animals, but there are several ethical reasons, as I said. So you won't get your studies cleared so fast for anything. So, and when you use radioisotopes and also radiation dose, safety logistics in higher animals is also a big challenge. And handling of injected animals, those becomes very difficult. So you have to have a dedicated animal scanners for rodents. That was the need, and this goal started maybe last 2 decades. So these are some advantages has been mentioned here. So molecular and functional readouts we can get very easily. Then high sensitivity and signal recognition. So any radiation tools, when you use it, it gives very high sensitivity in signal recognition. As you must have seen carbon dating technique. Yeah, it tells you about the age of any rock you hit, they will tell you the age because they are very highly sensitive. There are probes which are having a high affinity and they are molecular probe. Then unlimited depth of penetration. Like if you go for ultrasound, you cannot do ultrasound for all organs. Yeah. Because there is a issue of depth of penetration. But when you use radioisotopic agents, you can really very well do that because there is no question of any depth of penetration. And most importantly, you can do an absolute quantitation. Yeah. So target to non-target ratio. All those things, even the percentage uptake in that organ. So those things can be really quantitated very nicely using this kind of modality. So if you see the conventional way, what we do usually, we do a biodistribution study, we sacrifice the animal at different time points, still we miss some time points. But when you use these modalities, let's say I am developing an animal model and that progression of the disease also can be validated with the same system. When that model is ready for intervention, you can do a therapy or whatever is your intervention is, and then you can test also a response to the therapy. Yeah, how it responds non-invasively. So you are using a single mouse for disease modeling also as well as response monitoring also. So it saves your time, it saves your animals, and the data outcome from this will be much better because there is no variation based on the animal to animal system. Yes. When you use number of animals at different places, there will be a variation, and those factors are out of your control. Yeah. But here it gives very precise data about it. So longitudinal follow-up of disease process and response to new molecules is possible in a single animal. So this is the drug discovery pipeline. You have seen that the lead identification is there. Then bioinformatics, in silico, a lot of techniques are there, softwares are there that can give you the in silico analysis of many things. So structure-based drug designs are there, and then you go for in vitro studies, basically the biochemical assays or cell line assays, and then it comes to a preclinical evaluation, and this has seen as a most important factor in a drug attrition. When your preclinical data is not robust, there is a chances are there that drug might not really work the way you are having a hypothesis about it. And this is also important if that drug is not going to work. If I come to know early, so I can just leave that drug and work on some other target. So it saves your time, it saves your resources, and basically you go with a very right kind of a molecule. So this is how and this whole process is like maybe 10-12 years of process, and now it has come down to 6-7 years. Yeah. And COVID, we have proved that we can even produce a vaccine within a year. Yeah. So basically, it's a mindset, it's a willingness to do the things and do the things appropriately. So this particular imaging modality started from 2008, and you can see how those modalities have developed. So when earlier when they started, they were having very crude methodologies to develop this kind of modalities. And now there are very sophisticated modalities are there. And earlier there used to be a 2-3 kind of modalities together, and now the single modalities are there, and the exchange of fusion imaging they call it, as a multimodality imaging we call it. So we can fuse the data from different varieties of systems, and we can see the image as a single image. See how this systems looks like. It's a very small kind of a compartment now. Earlier there used to be a huge kind of machines, now they are compacted in a very small way. Workstations are there and you can get very nice kind of high resolution images. So this was a tri-modality system where PET, SPECT, CT all are together. So in a single scan you get so much of information. But there the issues came later that okay, my PET is not working, I have to save it for maintenance. So both the modalities are down. Yeah, I can't use it. So people are developing now the separate modalities and you can fuse the data out of that. So we also got, in 2007 we got a tri-modality PET, SPECT, CT and all. And now when we got the new systems, now we are getting the different isolated systems. Yeah. So we don't want, fused kind of systems together. So I'll give you some examples about the positron imaging tomography. So tomography means 360-degree kind of viewing. Yes. Seeing that object in a 360 degrees, and that we call it a tomographic imaging. So that you get a 3-dimensional picture of that organ system. So these are the technical parameters about the whole system. This is how the array of detector is there. Where the detectors LYSO and LGSO crystals are there, very highly sensitive for the positron gamma emissions. And this is how the reactions takes place when you inject a positron emitter into a subject. So there is a reaction called annihilation reaction. Probably when you think about your 10-12 standard days, annihilation reaction is the one which can give rise to a 511 keV gamma photons in equal and opposite direction. So subject is here and you get 2 photons in this direction, and this photon should reach to the detector in some time that called as a time of flight, and it usually 8 nanoseconds. So if any photon reaching to that site after 8 nanoseconds, it means that the origin of that photon is not the same. So that's how these algorithms are developed, and that's why the systems are very sensitive to the resolution. So very high resolution systems are there. Because there is a digital collimation, we call it as a digital collimation, so it is there. These are the phantom which are used for find out the resolution of the system. Here you can see that this is 0.7 mm, then 1.2 mm also. We can resolve very well with this imaging system. So how is the study protocol? Study protocol is you have to develop an animal model system, then radio label the drug which is of your interest, then anesthetizing the animal when you do a scan, then do a imaging at different time points, and then image visualization, analysis and quantitation of those images. So this is how this protocol works. And this animal handling features are very important because all these animals are under anesthesia. So they should not move even a slight, even a breathing rate if it is going too high and low. So that can cause artifacts in your image data. Because this is a high resolution image, and in that even a small movement can cause a bigger artifacts. So that's why there are pre-plumbed anesthesia lines are there, so that the animal is throughout the process, animal is in a general anesthesia stage. So there are integrated physiological monitoring systems are also there. Like ECG getting is there, respiration, you can monitor about those elements, temperature you can monitor. And gated studies are quite important in some of the cases, like if you are looking for lesions in a lung, so you need to do a gated respiratory studies. So there, this integration of physiological monitoring systems are very important, so that you can get some better data and high resolution data. Then even you can do a dynamic scanning also. Suppose that drug immediately after injection, how it goes into a body. So it goes to a bloodstream, then after bloodstream where it goes, how long it stays, and how the metabolism occurs of that drug, whether it's through a renal way or through a hepatic way. So all those parameters are very much important when it comes to a new drug. To understand that drug. So let's see which are the positron emitting radionuclides, because we have to label those with those isotopes. So here you can see that there is a range of radionuclides are there, which have a ability to emit the positrons. So this F18 is highlighted here. It's a fluorine 18, and it's a cyclotron produced. Cyclotron is nothing but the small reactor, and which has potential or which has capacity to produce isotopes of medical importance. So it is a very kind of a high cost equipment, and in Mumbai we have only two cyclotrons, three cyclotrons, two are commercial and one is with government sector. And F18 is produced there every day. There are other isotopes are there, but if you see there are half-lives, like C11 is a very good isotope where you can label amino acids with that, but half-life is just 20 minutes. You can see O15, it is 123 seconds. In just a 2 minutes half-life is there here. So many times it is produced and directly injected into the body. Like O15 labeled water is used to find a blood flow to the various organs like brain. So there this isotope is very important, O15. But the logistics never always in our favor. Copper 62 is there. Copper 64 is 12 hours half-life. And you can see the ideal 124, it is very good to label monoclonal antibodies and some of the proteins. And half-life is also 4.18 days. So it can be produced, it can be labeled and shipped wherever it is required for your studies. So this half-life is one of the most important criteria. So we get our in our facility all those positron emitters we get from Parel. So it comes from Parel lab. Mainly we get F18 labeled compounds. C11 they don't really produce much. But in F18 also there are various tracers are there. So I will just give the brief about those things. So if it comes to a cancer or any kind of disease processes, this is a picture of a cell, and you can see there are several targets are there. And if you see that there is one kind of a tracer is there for each and every target to quantitate that target. So it's such a wonderful tool. If you have that target, and if you know how to quantitate that, your data, your outcome of the results will be very proper and reliable. So you can see FDG is there, which is targeted to the glucagon receptors, which is basically for a glucose requirement of the cells. Choline is there. Then you can see amino acid targets are there. F-DOPA. Then FLT is there. It is nucleosides to thymidine kinase target is there for that. So the proliferation rate of those cells can be measured. Here Fe is there. Estradiol. So it is a estrogen receptor target is there. So here we can also check how the FES receptors are there. So it's particularly useful in the cancers which are influenced by the hormones, like breast cancer in females. They have ER/PR positivity is there, or negativity is there, and that always gives us idea how the disease is going to be there, whether it will be very highly aggressive or not that aggressive. And on this target basis, there are new therapies are also available here. So if these targets are positive, then the therapies can be recommended for those patients. So this is what is the reactor medical cyclotron. So it's a very small machine that we constrain our real big reactors. And but this requires all regulatory permissions and all that constructions for that very different way. These are the tracers actually available based on the F18, Euro 18. So FDG is the one which gives you idea about glucose metabolism. NAF is a sodium chloride which can be used for bone imaging, particularly to know the metastatic lesions in bone skeleton. Then F8 is fluorothymidine for proliferation. Meso is for hypoxia imaging. So many cancers are there when they proliferate, their core is hypoxic core is there. And there are few targeted therapies are coming up for hypoxia. So in that situation, this meso is very useful. Similarly, FASA is there. Uroazomycin arabinoside, that is also a hypoxia imaging agent. FET is there for brain tumors, because brain tumors you cannot use FDG because brain also needs a lot of glucose. So you cannot resolve the brain tumor with FDG. So you have to have some other agent which can be used. So FET is the good kind of alternative for that. So this is again a whole preclinical protocol how it works. So after radio labeling and all evaluation in small animals, then large animals, and then it goes for human clinical trials. So when it comes to the radioisotopes tracers or therapeutic agents utilizing radioisotopes, it goes to a radiopharmaceutical committee, RPC they call it, and that committee gives the clearance for the human trials. So all the radioisotopes used in this are in a tracer amount. Even the drug which is used, radiopharmaceutical, in a tracer amount. So they doesn't have any kind of ability to generate a physiological effect. So that will only used to evaluation of the system, but they don't have ability to cause any kind of physiological effects on body. So that's why they call it as a tracer technique, and that's the reason they are very safe that way. When radiation comes, we always remember it is hazardous kind of thing. We always have in mind that radiation is harmful to us. Yeah. Yes, radiation is harmful, but there is a regulatory agencies are there who are looking into how much our exposure is there for the radiation. Yeah. So when we do a radioisotopic scan, and if you compare that with the X-ray imaging, so X-ray imaging gives you much higher exposure than the radioisotope scan, though it is administered into a human or animal subject. Yeah. So in COVID-19, we must have seen several times debate on whether the high resolution CT, HRCT is required or not, and how often you should undergo. Many patients used to say, nothing is seen in your chest, then go for high resolution CT. But high resolution CT gives a extraordinary high resolution, high exposure to the radiation to the body. Yeah. And we really don't know what will be the effect on our biological cells in future. Because there are two types of effects are there, acute and chronic effect. Yeah. Stochastic and non-stochastic effects. So stochastic are always an acute effect, we can always get reaction after a week and all, we can understand that. But the one which will come later, it is beyond our control to even understand why it is happening to us. So HRCT was not recommended unless and until it is medically required for that patient. Yeah. So similarly with X-ray, you cannot just go and say that I want to do my chest X-ray. So they will always ask what is the indication for that. Yeah. And when we are sitting here, still the radiations are passing through our body without our we noticing it. The rays coming from Earth crust, solar radiations, they are continuously passing through our body. We even don't get any kind of understanding about it. So we have to, whenever there is a radiation area is there, we have to really understand, we have to respect that yes, radiations are harmful, we should not go unnecessary to that area. We should not get ourselves exposed to that area unnecessarily. So there are some of the principles are there when you work with radiation. One is protect yourself, but not going there. Yeah. But who can protect? Those who don't have work in radiation facilities, they can say, okay, I am not going. But who is working there, he has to go there because it's a occupation is there. So occupational for hazard, he has to follow certain practices. So this is a lead shielding is there. The shielding is most important thing. Whenever radioisotopes are there, shielding has to be there properly. Time. You are in that radiation facility has to be minimum. Your work is done, you go away from that place. Yeah. Then distance from yourself and the radiation source, that has to be maximized. Yeah. So you use tongs, forceps, and increase the distance while working. So these are the basically, even you can see here in animals we use a microcurie level kind of a drug. Yeah. In humans we inject it really pills, but in animals, mouse, we use like 30 microcuries. It's such a small dose is there, but still at our place we use the syringe sheet. Whenever syringes are there, they are always in a sheet. Yeah. And safety hoods are there. Because some of the radioisotopes, they are volatile in nature. Like if you see iodine, it's volatile in nature. Yeah. So you have to have a safety hoods when you dispense them. You make the smaller doses. So you can see here, this is a syringe sheet. So safety is the most important thing when you handle the radiations. Yeah. Then challenge comes techniques. Your tail vein injection. It is very difficult to locate a tail vein of a mouse, which is just 20 grams, and there are tiny veins are there on his tail. It's very kind of a struggle to find a tail vein. Yeah. You all must have struggled in your research days and all. Now you can very well tell to your student that okay, I have done in my college days like this, my PhD days, and you do it. So any skill, when you repeatedly do it, you will be better in that. Yeah. So we have to have 1 or 2 person in our facility who are skilled in giving tail vein injections. Because when you use a radioisotope, you cannot afford to go any single microcurie outside the blood pool. Yeah. Because it is going to give your quantitation in a very wrong way. Yeah. So it has to go into the blood pool. So you can see here, our technician has injected into this, put the needle, and you can see the red coloration here. Immediately the blood comes, and then you are sure about it, and you can push the radiopharmaceutical further. And you can see here, he is using the shield also for that purpose. Yeah. When he is injecting that, here it is only the saline he will push, then put the radiopharmaceutical, and then again he will put the saline to flush all the remaining tracer into that tubing. So it's a F10 tubing is there, and these chemicals are made in our lab. So now let's see the imaging how it comes. Here you can see the FDG image. This is a whole body survey of mouse in one single study. Here is the tumor xenograft is there. You can see that the uptake of FDG, where it is intense, it is red hot here, and where it is blue, there is no uptake of that tracer here. So you can see here that uptake is not uniform. Why it is not uniform? Because tumor is not a uniform kind of a system. It's a very highly heterogeneous kind of a system. There is a proliferating core is there, periphery is there, inside core is hypoxic, then sometimes it is necrosed, depending on the malignancy rate progression of those cells. So my agent is, let's say it is targeting the hypoxia. So in imaging I can find it out whether it is really going to the hypoxic lesion or not, that agent, and whether that area is getting reduced after therapy. You can here very well see this. This is a ABCD scan, progressive disease, and 0 day, 17, 23rd and 38 day scans of FDG. Here there is a very uniform kind of uptake is there when tumor is small. But then you can find only the peripheral area is having uptake and core is totally necrosed or hypoxic. So if your target, assume that it works on a periphery, so you can even quantitate what is the uptake in a periphery, what is the uptake in core. So such a precise quantitation you can do it. In normal way, in conventional way, how we do our these studies, we take a vernier caliper and we check what is the regression of the tumor. But here you can see inside the tumor what is happening. Many times tumor might not shrink, but definitely the core is getting damaged. Or core cells are getting killed. So these are the things which is very important. So these are the examples how the images are seen and how we can quantitate them. So you can see this is a xenograft model. Xenograft means it is a cells, human cancer cells are injected in a mouse which is immunocompromised and it is given in a flank region subcutaneously. But when it comes to orthotopic model, orthotopic means the organ from where that cell line is generated, like it's a liver tumor. So you have to inject it to a liver itself. The reason for that is every tumor requires a normal stroma to grow. So breast cancer put in a flank, it will take some time to grow there because there is no normal stroma or normal environment for that. But when those cells are put into the fat pad of mammary gland, definitely it will grow much faster, and that is the right way to do the thing. So this was our study to establish orthotopic syngeneic rat model and its validation with microCD. So you can see here first week, second week, how the tumor is growing into a liver, and also the gross after the sacrifice. Many studies they are related to targeting to this. You can very well do the timely imaging. So this is another example of some study in a xenograft model, and you can see here the intensity of the FDG in that, and when it was treated, the intensity has been reduced. So these are the kind of visualized images, and these tracers are used for the human studies also. So the reproducibility is much better. There are certain tumor models are there, that is small mice. It is give medulloblastoma, and here you can see the medulloblastoma lesions are there in a brain. So this is not possible by any other technique. Even on X-ray you cannot really see, but the FLT imaging you can see the things. So this is another example of FLT image. This is a normal scan. So there is a normal distribution of FLT. So it doesn't go to the extensive to any organ, but when there is a tumor is there and tumor is progressing very fast, FLT uptake is always very high in those cells, because this is a marker for DNA synthesis and cell proliferation. As I said, the sodium fluoride is used for a bone metabolism. So there are many diseases are there, many cancers are there where the bone metastasis is there. And in those metastases, there are micrometastases also. So to know the number of metastases as well as micrometastases, this FLT is used. NIF is used, sodium fluoride. can find a standard uptake value in tumor models. Here is the xenograft tumor, and we can find that SUV, and that SUV can be used for comparison in our whole study. Even in human patients now, SUV is a very commonly used to find out the response to the treatment. Yeah, this is one of the most important criteria. So I'm just going to skip some of the things. How much time do I have? We have how much? You can take.
Can go up to 30 minutes.
Very good. Let's skip some of the slides here of PET imaging, but we can because SPECT is also there and CT is also there. So you can. I will just go very fast on those. So you name the kind of a target and you will find there is a tracer is available for that. This is what is the kind of a beauty of this system. And many times you can use a multiple tracer in one system. Yeah. So there are examples where multiple tracers can be used. Like in some case I want to use FDG as well as FLT. Yeah. And then there are subtraction methods are there where you can reduce or subtract the data, that image from the original one, and you can get very sorted image of one isotope. Yes. And that is very important in when, because FDG is a non-specific oncological tracer. Yes. So it can go to the inflammation also, it can go to the wherever the glucose uptake is high. So in such cases, dual isotopic imaging is very important. Even people are using now the PET-MR hybrid imaging. So in this magnetic resonance imaging (MRI). So these are the modalities now. Single is not useful, so you do a hybrid imaging. So PET-MR, PET-CT, these are very common kind of thing in translation now. So you combine the information from both the modalities and you try to understand the behavior of that disease. Yeah. So that your target can be very well defined. So we go to the SPECT radioisotope. In SPECT, you must have heard about a technetium all the time. So technetium is a very, kind of a very dynamic kind of a isotope where it has an energy of 140 keV and half-life of 6 hours. Very suitable for imaging as well as, radiation safety purpose. Thallium is also there, and technetium, can be labeled with several molecules because of its very dynamic chemistry. Thallium is the one which is similar to the potassium ions. So it is used for cardiac imaging, myocardium imaging. Indium is there, which can be used for radiolabeling with RBCs. Iodine 131 is basically a therapeutic isotope, but any iodine molecule can be labeled. Without labeling can give you the good images of thyroid gland, and also the monoclonals can be labeled with that. So whenever radioisotopic imaging comes, energy of that isotope and half-life, these are the most crucial factors. With technetium, there are so many tracers are available in human clinics. They do it all those scans very routinely, and same tracers we can use for animals. So your biomarkers are same. Reproducibility becomes much easier when it goes to the routine clinics. So this is about the instrumentation part. So these detectors are also very kind of highly sensitive. Conventional gamma cameras have a photo multiplier tubes and all, but that have been totally replaced now with new technologies, and they have become more compact now. So there is noise level in your images has reduced a lot. So it's a C-Z-T gamma cameras are there, cadmium, zinc, telluride, highly sensitive, and they have ability to even resolve different energies. So same thing here, multiple isotopes you can use in a single animal, and you can collect the data of different isotopes separately. because of the energy resolution abilities of that detector system. So proof of concept is one of the most important things when we do our research. So this study was done for just to do that proof of concept validation, and this material or nano assembly which was developed, and the aim was to target it for the lung cancer. So how long does it stay in lungs? That was the main issue, and that issue was resolved using this kind of radioisotopic technique. So what we did, it was labeled with technetium, and the aerosols were generated and they were given to the animals. And then the images were taken at different time points like 0, 1, 2, and 4 hours. So you can see at 4 hours till 4 hours, there was accumulation of that particles or nano assembly was there, and that was releasing the drug. So the drug residence time in that organ can be very well identified using this. So this is how we can validate we can do a proof of concept study. Here again it is very clear that you can see that drugs comparison with the drug loaded lipid assembly as well as blank lipid and drug solution. You can see here it is just stuck into the tracheal tree. So this was another kind of experiment we did for liposomal system which was targeting to the liver cancer. And you can see here after injection almost more than 90% goes to the liver itself. Here. And this definitely then it will stay there and release the drug there. So you can get very nice images and you can see the liver there in that completely having the uptake of that liver compound. So DTP and meglofenin, these are very much important to see the toxicity. So renal toxicity and hepatic toxicity, before it is evident on biochemical test or any radiography or ultrasound, you can find it out on the basis of this DTPA scan or meglofenin scan. This is what is the functional parameter of this any kind of renal or hepatic organ. And in our one of the study, the Ayurvedic drug compound called as Vasmas was evaluated for this, and we could able to see there was no on histology, no changes were there or damages were there evident on that, but on scan we could able to see there was a trend which was saying there is some abnormalities there in the renal function. So this becomes very much important when we are looking for toxicity at very early stage. So this is the dual tracer imaging. Even PET and SPECT probes can be used together. And now recently we have done one study also on this technetium labeled the drug as well as FDG scan was done in one animal recently. Very interesting data can be generated. We go to a third modality which is a very extraordinary modality nowadays, and it's a micro CT. As name suggests, micro, it gives you the micron resolution. And the system which we will be going to get in our lab now, it has a resolution of 3 micron. Yeah. So the bone trabeculae, counting of the trabeculae, all those things will be very easily possible. Even micrometastasis in a bone. Then calcification in some of the organs. Yeah. So those things can be quantified very well. So this is how micro-CT analysis can be done, where you can use. Basically, this is useful for the studies which are related to the bone damage or osteoporosis. Yeah, osteoporosis is a very important issue in females, and that is not yet resolved. It's always very big concern after the postmenopausal period or 50+ age. Micro-CT plays a great role in lung metastasis also. Here you can see that the number of metastases in lungs, that also you can count. Yeah. So if you can see here, the lungs looks like, it's a air only. So there is no kind, it should look like a black only. Okay, on X-ray. And the bones look very white, and this soft tissue looks grayish. So this area, you can see there are the soft tissue, densities are there. So these are nothing but metastatic lesions in the lung. So this study we did for one pharma company, and that drug was highly effective as per their kind of studies against the metastatic lesions in the lungs. Yeah. So they wanted to prove it with microCT, and this study was done. And then that CRO, they have sold this agent to a one pharma company at a very high price, utilizing this data that yes, it reduces the number of metastatic sites in lung. So very useful tool. Other area which is important is the bone implants. Yeah. So these implants, now there is a lot of refinement in implant technologies have come. Earlier there the metal used was, kind of a very crude metal, then titanium has replaced. Now titanium with some new technologies, the different surfaces have been used. Then now the stem cells are used for this purpose. So stem cells coated kind of implants are there. But the most important part of these implants is the osteointegration of that implant with the native bone. Yeah. And for that purpose, microCT really gives you a very good kind of data on this. So this study we did for the dental implants. So this study was done in rabbit, and you can see that that the screws used there as an implant are integrated, and there was a much better integration than your routine practice. Whatever the standard care is there on that basis. You can also use these technologies for contrast development or contrast imaging. There are a lot of scope is there for MRI contrast as well as the CT contrast agents. Their main issue is that CT contrast agent doesn't stay in a system for a longer time. So the blood residence time is very low. So you inject it, if you delay the scan, everything will be seen in bladder. It goes so fast. So there are a lot of research groups that are developing new contrast agent where the residence time in the blood is very high here, so that the soft tissue contrast in organ of interest will be better. So for that, microCT is very much important. So another scaffold was implanted in a rat. It was kind of a flat bone scaffold. So this is one more area which can be used for this purpose and of your interest is the delivery route of different few routes. If you want to give that drug in a very different route, so like nose to brain targeting was seen here. So this is only can be validated using such kind of tools. Then you can see here, this is the orthotopic brain tumor, and which has been seen at different time points how it is growing. You can see how nicely you can visualize the brain tumor in mouse brain. So the similarly the regression can also be seen like that. It gives you a clear-cut idea about how your drug works. So I will just give this few slides. So even without cutting a bone, you can enter into a bone and see is there any kind of lesions are there here. So if there is a model of, let's say, bone cancer in fibula or tibia, so how much percentage is there inside that bone can be calculated so well. So it is called as a virtual ex vivo endoscopy. So all these modalities, when we use, the tracers used are same. Okay, you can see here mouse, rat, preclinical level, tracers are same, even clinical level tracers are same. So the translation is very fast and very robust here. So that's why these are also called as the translational imaging modalities. And not only in cancer, there is so many applications are there from these modalities. And even this is in a medical field, but even non-medical field also there are several applications are there for microCT and other modalities. And this is what is the future, because of new libraries, new molecules, new targets. So many targets are there. So we want to have system like this where you can scan those targets so quickly and decide whether it will work or not. If you want to fail, you fail fast. Yeah. If you are promising, then it should be taken fast to a clinical level. So such kind of systems are being developed where we can scan it very quickly and very fast. So challenges are always there and they are going to be there. So this is the list of challenges where dose administration is a big challenge. Volume, radioactivity, half-life, then distance. Radiolysis is the thing which is very important when you handle high amount of activity. Then anesthesia, that is another issue. Radiation protection, all those things are there. And on top of that, funding, whether we have funding or not, that is very challenging part. So with this, I end my talk here. I will be very happy to receive, any kind of questions are there. And even if you remember your questions later, you can email me also. I'll be happy to answer. Thank you very much for your questions. I'm feeling very uncomfortable to sit there. Yeah, I will sit below only. Yes.
Thank you very much, sir, for the insightful session. Now I request Ms. Mamta Meena to summarize the session. Thank you, Pradip. First of all, I would like to thank Dr. Pradip Chaudhari sir for giving us an opportunity wherein we could have an interactive session about how medical imaging has taken its face even in the preclinical trials. Sir started the session with giving us an overview about the preclinical experimentation, what was the history, where it started from, and then he went on ahead with tracing it back from the conventional techniques where it was used for the human diagnosis to the preclinical testing in animals itself. Sir also took us through the different types of analytical techniques such as PET analysis, the computer tomography and micro-CT analysis, and also gave us illustrations examples where we could try to identify how the radiopharmaceuticals were traced, and as well as helping in understanding the metastasis in different forms of cancer. Yes, sir did say that these methods are very costly, but they are also better if you want to have a full-fledged knowledge about whether the disease is progressing, how it is progressing, and whether your pharmaceutical agent is working or not. Plus, sir also gave us an idea about the challenges which are being faced right now in terms of radioactivity, the radioactive safety, the challenges in maintaining protection for the personnel who is handling them, as well as the future scope of these medications. Plus, sir gave us an idea about the applications, not just in cancer, as well as in agricultural biotechnology, and we as pharmacists can also try to identify whether we can come up with any contrast agents and as well as work in this field. So thank you so much, sir, for giving us an idea and the kind of work which you are doing at ACTREC for also this I think might have given an opportunity for the delegates as well to think in that direction of not just having preclinical studies for disease progression over conventional methods. We can even try if we have funding for this method as well. Thank you, sir. The session is now open for question and answers.
Sir, actually, can you throw a light on what is the difference between a tracer and a marker?
Tracer and a marker, yes, yeah. Basically, these are the synonyms. Yeah, tracer, then marker, then probes, molecular probes, targets. These are quite synonyms and they are used, exchangeable anywhere. Yeah. Right, absolutely. But tracer is the terminology was given basically when it started, this whole story started, because they are used in a very trace amount. Yeah. And it doesn't cause any physiological effect on the body. So that's why they are called as a tracer kind of techniques.
So I have one question pertaining to in silico methods like bioinformatic methods. How these methods are helpful in the toxicity testing because you can predict RPR, ADME also, PKPD also, so toxicity. How they are really helpful to carry out in vivo testing.
See, any kind of a in silico test that depends on how robust that algorithm is. Yeah. And there are now new algorithms which are there with industry. Probably we are also not exposed to those things so well. Yeah. So those things can predict very well. Yeah. And there are drugs which are coming from in silico studies now.
So because you said carcinogenicity toxicity, so that can be predicted at earlier stage with this software, right?
Absolutely, but that depends on how much is there in your database. Yeah. So if database is really very strong and have a lot of kind of disease drug libraries are there, molecular libraries are there, then it can predict very well. Yeah.
So next question. I would like to understand whether this imaging technology can be used to check whether my drug will cross the blood-brain barrier or not. In quantitative way or in qualitative way, sir?
Absolutely. Yeah. See, what happens, there are diseases are there, like meningitis, okay, where this barrier is damaged or disturbed. Yeah. So there is a singular technique is there. When technetium perchloride, when you inject, it doesn't have ability to enter into your brain. Okay. But when barrier is broken, it will go and reach brain. Yeah. So on imaging, if you can see the brain, that is indirect indication that brain BBS is damaged. Yeah.
So in a normal animal, how can we see? It is not damaged.
So it won't go there. Yeah. So what you can do, you can use a lipophilic agents where they can pass that barrier very well and reach to the brain. So the agents which are very good, like there is an agent which is very good for the brain tumors, but it is not crossing the blood-brain barrier. Then you encapsulate it in a form which is highly lipophilic, and then it enters into your brain. So likewise, you can do the things.
That drug release I can quantify.
You can do that. Yes. Drug release is always indirectly, it is proportional to the uptake of that tracer in that organ. Yeah.
Thank you, sir.
So, thank you very much for all of you. And any questions are there, please write back to me. Thank you very much.
I request Dr. Anita Aire Madam to felicitate Sir with a memento and certificate as a token of gratitude.
Don't you think too many formalities are
Aparna madam, announce for group photo.